Je vais donner ma conférence tout à l'heure en anglais, mais je vais me présenter d'abord en français, juste pour vous donner l'histoire de ma carrière. Je suis d'origine suisse, mais je suis venu en Angleterre pour faire mes études. Et puis après, j'ai commencé à travailler pour le service public de santé en Angleterre. Et ensuite, j'étais prof dans une école polytechnique en Écosse. J'ai fait un retour en Suisse pour travailler dans l'industrie pharmaceutique dans ma ville d'origine, c'est-à-dire Bâle, dans le nord de la Suisse. J'ai travaillé pour Ciba-Geigy, qui est maintenant fusionné avec Sandoz pour créer Novartis. Et puis après, j'étais prof au collège universitaire de Londres pendant 8 ans, et ensuite aussi pendant 8 ans à l'université de Glasgow. Finalement, pour finir ma carrière, j'étais le chef du centre de compétences méthodologiques pour le Luxembourg Institute of Health au Luxembourg. Et puis, je suis à la retraite depuis 2 ans et demi, mais je continue à faire de la recherche et puis pour donner des conseils statistiques pour l'industrie pharmaceutique. Donc, ça, c'est ma carrière. OK. So now I shall switch to English. I'm going to talk on the subject of statistical pitfalls of personalized medicine. And this is a topic I'm interested in because although I believe that it's worthwhile trying to personalize medicine where one can do so, I also think that people misunderstand the statistics and they consider that the scope for doing so, the possibility to do so, is much larger than it is. So first of all, thanks to Roche Georgi for the invitation and to Alain Emmerich for technical support. And just to mention that the research on which this is based was funded by the European Union's 7th Framework Programme as part of the IDEAL project. So I'm going to give you a number of quotes from eminent people on the subject of personalised medicine, and they are all very enthusiastic. The first one is by Sir Richard Sykes, who was at one time the chairman of GlaxoSmithKline and later of the merged company Glaxo Wellcome, but also the principal of Imperial College in London, one of the great universities in the United Kingdom. And this is what he says. We'll have genetic tests. Individuals who will respond favorably to the drug candidates based on their genome will be identified, and we'll have smaller, more effective clinical trials, and individual patients will be targeted. And this now is nearly 25 years ago. Here's a leading researcher. This is Francis Collins, who was at one time head of the NIH, National Institutes of Health, United Kingdom, in the USA. I don't know whether he still is. And this is what he said. Not only will genetic tests predict responsiveness to drugs on the market today, but also genetic approaches to disease prevention and treatment will include an expanding array of gene products for use in developing tomorrow's drug therapies. And that's from 1999. Here we have a previous Prime Minister of the United Kingdom. This is David Cameron, the person that many people regard as being responsible for the whole Brexit disaster. So this agreement will see the UK lead the world in genetic research within years. I'm determined to do all I can to support the health and scientific sector to unlock the power of DNA, turning an important scientific breakthrough into something that will help deliver better tests, better drugs, and above all, better care for patients. And to continue the list of important actors, if you like, in this drama, this is the world's leading regulatory agency, or it likes to think it is. This is the Food and Drug Administration of America, and this is a particular report issued in 2013, paving the way for personalized medicine. I'm not going to quote from that now, but we'll come back to it. And here we have the leading evidence-based medicine organization, the Cochrane Collaboration. I, by the way, am on Twitter. I'm rather addicted to Twitter, and this is something I saw a few years ago appear on Twitter, which attracted my attention. Featured review, only 10% of people with tension-type headaches get a benefit from paracetamol. Sounds shocking. Only 1 in 10 people benefit. And when you look further, these are the figures it's based on. 59% had no or at worst mild headache after 2 hours when treated with paracetamol, and 49% had no or at worst mild headache after 2 hours when treated with placebo. So the difference between 59 and 49% is 10%, therefore 10% benefited, and if you divide this into 100, 10 into 100, you get 10, and so that means that only one extra patient had a benefit when you treated 10. And this is based on a review of 23 studies and 6,000 patients. By the way, this particular number of 10 is referred to as the number needed to treat. The idea is it's how many patients you would have to treat with a new treatment, say paracetamol, compared to placebo for one to benefit. And this is the financial press. This is from 3 years ago, claiming that dramatic advances in our understanding of cancer biology and genetics are helping to deliver an era of precision cancer treatment. So this is another extremely optimistic claim about what's happening. So here's a warning for you. We tend to believe the truth is in there. We tend to believe that somehow there is something that we can explain if only we do more experimentation. The problem is sometimes it isn't, and the danger is that we'll find it anyway. And I hope to try and persuade you in the rest of this lecture that the scope for personalised medicine has been vastly overstated. So let's have a look at the FDA on non-responders. This is from that FDA report which I mentioned before. And I'm going to highlight the particular condition of migraine. And here what the FDA gives you is the proportion of persons by particular drug, or by particular, disease, who don't respond. And what they say is in migraine, 48% of patients don't respond. By the way, on seeing this particular diagram, I immediately was somewhat suspicious because if you look at the bottom of the diagram, you see that 75% of patients don't respond on cancer. That's a very strange statistic to give because actually cancer is a mixture of diseases. The response rate in lung cancer is very, very disappointing, but the response rate in testicular cancer is very, very high. And so it's rather strange to see a mixed figure like this. We already personalised treatment in that sense. So the question is, where did the FDA get these figures from? Well, they give you an indication. They cite this particular paper from Trends in Molecular Medicine. And here's a strange thing. The FDA report is from 2013. To tell you what percentage of patients don't respond in 2013, they are citing data from 12 years earlier. But surely in those 12 years there's been some particular progress. So why was it necessary for the FDA to go back 12 years to get these particular figures? And the reason is, in my cynical opinion, that there are no figures. There are no reliable figures, and they were told to try and look for something, and they looked and they looked and they looked, and they found this particular paper. By the way, if you have a look at the paper, and if you have a look at migraine, you will see that the figure for migraine is 52, whereas the FDA's figure is 48. That's because in the original paper, they gave the percentage of patients who responded, and in the FDA report, they concentrated on those who didn't respond. And 100 minus 52 is 48. Okay, so there's this paper in Trends in Molecular Medicine 2001. But the question is, where did they get these figures from? Well, let's go back to the FDA. The FDA got it from Spear, Heath, Chiozzi and Huff. But where did they get it from? Well, the answer is here. They got it from the physician's desk reference. And at this particular point, having traced back all of these figures, because I'm a nasty person, when people tell me something, I like to say, how do you know this is true? And dig and find out. And this is where I realized that what we had here was zombie statistics. This is not to be rude about the paper by Spear, Heath, Chiozzi and Huff. There are many interesting things in this paper, but this particular aspect of it is really not a solid basis for judging response. Because what they basically said was, we looked at the physician's desk reference. What is the physician's desk reference? It is itself a secondary source. It's a compilation of different papers and different results throughout, which is given as a gift, or the physicians can buy it to help their prescribing. This is not a reliable scientific source. It's a secondary source. And furthermore, they don't even tell you how they managed to get these figures, physician's desk reference. So what we have, in fact, is an example of a zombie statistic. A zombie statistic is a statistic which somebody says on one particular occasion, then somebody else cites it without really looking at it, and the statistic then cannot be killed. You try and kill it, it just comes back, and people will say, don't you know that only 10% of people respond to paracetamol if they have tension headache? These are zombie statistics. They're ugly, they're evil, and they refuse to die. And what I maintain is that even if the figures were right in some numerical sense, but I don't think they are, they wouldn't mean what they are assumed to mean. We need to do something. And one piece of advice I would give you is, whenever you hear anybody talk about the brave new future of personalized medicine, ask them if they know what response rates are at the moment in various diseases, and if they know what sources they can cite. So now I'm going to show you how easily it is for a statistician to create data, to simulate data that would give you exactly the number needed to treat of 10 that you find in that Cochrane review, but in which every patient had exactly the same benefit. This may seem surprising, but it's actually quite easy to do. So what I'm going to do is I'm going to generate one random number. I'm going to generate a random number. It's a randomly, uniformly distributed between 0 and 1. Every value between 0 and 1, 0.3565, 0.2189, 0.9218, each of these values will be equally likely to be generated. And then I'm going to calculate pairs of headaches. I'm going to take minus the logarithm of this random number and multiply it by 2.97, and that will give me a placebo headache duration. I'm going to take exactly the same number and I'm going to multiply it by 2.24, and that will give me the paracetamol headache duration. Now, it would be lovely if in studying medicines I could simply rewind time and I could say, oh, this time I gave the patient paracetamol, but what would have happened if I'd given the patient placebo? It would be lovely if I could do that, because then I could really get to the bottom of causality. Unfortunately, I can't. With a parallel group trial, what I have to do is I can only have one of these values. I can only see one of these values. But when I simulate, I'm the god of the universe that I create. And so in simulation, I can actually do this. I can actually simulate what is called a counterfactual, the value that would have happened had I done something else. And I can then draw the empirical cumulative distribution. So here's a little example of how it works. Each of those points represents the two values that I could simulate. On the x-axis, so on the axis, just going to try and find a pointer. Excuse me one moment. On the x-axis here, I have the duration in hours of the headache under placebo. On the y-axis, I have the duration of paracetamol. Each of these points is a simulated pair. Remember, I simulated one random number, and from that random number, I calculated a placebo duration, and I calculated a paracetamol duration. And what you will see is that the placebo duration is always higher than the paracetamol duration. In fact, the paracetamol duration is always 0.76 of the placebo duration. It's the same proportionate amount of the placebo duration. In other words, the pain duration was reduced by 0.24, nearly 1/4 of what it would have been. Here, for example, is one particular patient. This particular patient on that headache would have had a duration of 3.9 hours under placebo. The patient had a duration of 2.95 hours under paracetamol, and that's the patient here, and these are all the patients. But the point about this is that in every single case, the patient has exactly the same benefit. Unfortunately, I can't see this particular diagram because I can only have one of the values. So now I've moved to simulating 6,000 values, 3,000 under one and 3,000 under another. This mimics more or less exactly what the Cochrane collaboration showed. And what I've managed to do by doing this is I've managed to produce a situation where 2 hours is the magical response value. The International Headache Association takes a response of under 2 hours as being, if the headache lasts less than 2 hours, then you responded. If it didn't, you didn't respond in this particular area. And it turns out that 49% of the patients responded under placebo and 59% under paracetamol, using this extremely stupid notion of response, which you will encounter over and over and over again in medicine. But actually, in my simulation, that's not what happened. Every single patient had their headache on every single occasion reduced by about 1/4. So I've completely mimicked the Cochrane claim, but in fact, there's no personalized response in this particular situation whatsoever. Of course, I don't know that this is true. It's extremely unlikely that it's true. But on the other hand, nobody knows that it's not true. And this is the problem. When people see with a particular clinical trial using a stupid definition of response that apparently 70% of patients responded, what they think that means is that 100%, 70% of the patients will respond 100% of the time. But another explanation is that 100% of the patients will respond 70% of the time. And you cannot simply tell from a parallel group trial which of those two would be true. So the lessons. The number needs treat of 10 is perfectly compatible with paracetamol having exactly the same proportionate effect on every headache. Nothing in the data we are given says anything whatsoever about differential response. And in general, the lesson is an NNT cannot tell you what proportion of patients responded. To think so is a straightforward conceptual mistake. If you think that, it simply shows that you don't really understand NNTs properly. And that unfortunately means about 95% of the people that use them. Claims regarding the proportions who respond based on MMTs are misleading. So what can I say about personalised medicine? Personalised medicine is a field that has proved highly productive. But what's it been productive in? Well, it's been productive in producing publications and grant funding. When it comes to actually producing treatments, less so. So here's an alternative approach to medicine. I'm going to give you the history of a great success story. And that story was the treatment of ulcers. So I myself now am age 67. During my lifetime, this was a particular disease whose treatment was revolutionized. And the story starts back in the 1960s with James Black, a great pharmacologist, working on H2 receptors at SmithKline French. And based on that research, by the beginning of the 1970s, they had a particular treatment that could block the receptors. This is burimamide. Unfortunately, it had other aspects that were not ideal, but the same team continued, and by the end of the 1970s, cimetidine was introduced by SmithKline French and already started to revolutionize the treatment of ulcers. A little later, in Australia, two researchers, Warren and Marshall, started looking for a possible infectious explanation for ulcers, the bacterium H. pylori. And Marshall actually infected himself to see whether he could get ulcers as a result, and he did. A little while later, Graham et al. Introduced a breath test which could check whether people were infected by this, and sometime later, omeprazole, even more powerful type of treatment, was introduced, and then a little while later, an antibiotic that was capable of treating H. pylori. And by the mid-1990s, the NIH had agreed the role of H. pylori, and the FDA approved the antibiotic. And then at the beginning of this millennium, Warren and Marshall got the Nobel Prize for their work. So what can we say about this? It's a great success story. There were 4 to 5 major innovations: H2 antagonists, proton pump inhibitors, the role of H. pylori, the C13 urea breath test, and together these transformed the treatment of ulcer treatment. And general surgery was largely wiped out. Before that, you used to have a bit of your stomach cut out to treat ulcers. You were better for a while, and then the ulcers came back. So this is a treatment for the many. Really, most people who have ulcers are treated in this particular way. Admittedly, the combination of the C13 test and the antibiotic treatment involves a degree of personalization, but medicine has always involved diagnosis. But now look at how the personalized medicine bandwagon misrepresented. So this is again from an interesting paper. It makes many interesting points. This is by Nicholas Schork. I'm just going to criticize one of them. This is what he claims. Every day, millions of people are taking medications that will not help them. The top 10 highest grossing drugs in the United States help between 1 in 25 and 1 in 4 of the people who take them. But this claim is based on number need to treat. This is my particular diagram showing the particular claim. And I'm going to have a look at one of them because it's closely related to the ulcer story I told you. It's Nexium, which is in fact a type of proton pump inhibitor. One of the 5 particular advances I referred to earlier was omeprazole, which is an example of a proton pump inhibitor, and Nexium is an even more powerful one. And the claim here is that it only helps 1 in 25 patients suffering from heartburn. So let's look at that claim a little bit closer. If we look at that claim, then what we find is that it's rather peculiar because, as I say, this is a highly successful class of treatment. The response is, if I have a look at a particular study that might in fact justify this, then it's by Labans et al. And the response is 92% in the case of Nexium and 88% in the case of the other treatment. But the other treatment, note, is not placebo. It's an alternative proton pump inhibitor. But this is the response at 8 weeks. And in actual fact, the response rises over time would probably increase beyond 8 weeks. So we have a situation very similar to the headache situation that I discussed earlier. And the claim that only 1 in 25 benefit is nonsense. It's quite possible that what happens is that in every single case, your ulcer is treated faster, or your heartburn is treated faster with Nexium than it is with the control treatment. So here's 2 extreme cases. Here's the EXPO study in which esomeprazole, which is Nexium, was compared to pantoprazole. And I'm considering 2 particular cases. Note, first of all, that for these 2 particular tables, we have the same numbers of patients in the margins responding. That's to say, for pantoprazole, 12.7% were not healed by 8 weeks, but 87.3% were. For esomeprazole, 7.9% were not healed, but 92% were. So in general, the healing rate is higher. And you can see that the same marginal figures appear here. But I've imagined that the same ulcer could have been healed under pantoprazole, or could have been treated with pantoprazole or esomeprazole. And I've imagined what possible tables I might see. And here are 2 extreme tables. I stress I cannot know the numbers inside these tables, but these 2 tables would tell very different stories. In this particular case, there is no patient who would respond on pantoprazole who did not on esomeprazole. What you find here is that actually 87.3% are healed under both treatments, but there's nobody that is healed under pantoprazole who is not healed under esomeprazole. It simply isn't the case. Whereas here, on the other hand, all patients who did not respond on esomeprazole would respond under pantoprazole. So what's the difference between these 2 cases? If this is the case, there simply is no point doing anything except giving everybody esomeprazole. If the first one is the case, there is no role whatsoever for pantoprazole. If the second is the case, there is a minority role for pantoprazole. If we could identify who the particular patients were who would be healed under pantoprazole and not esomeprazole, then in that case, it would be worth doing this particular diagnostic test. So here's a lesson for you. We've moved from finding highly effective treatments for most patients to trying to find expensive ones for almost nobody at all. So responder analysis, which is behind all of this, is the attempt to classify patients as responders or non-responders arbitrarily, often done compared to baseline. That's a bad thing to choose. Bad measures are often done. For example, we use percentage change from baseline. We have an arbitrary dichotomy. If your headache improves within 2 hours, then you responded. If it doesn't, you didn't respond. There's a linguistic confusion. We use the word responder and we think what it means is cause to improve, because that's what it usually means in everyday English. But actually it means was observed to improve. We don't know that the treatment caused the improvement. And in general, thinking that because something occurs after something, it was caused by it, is causal naivety. The English phrase is subsequence is not consequence. That which happens after is not necessarily caused by. And failure to replicate, if you want to exclude within patient variability as an explanation, you have to know how big it is. This involves measuring patients more than once. So this is from my paper on the subject. This is probably the most interesting slide in the whole of this particular presentation. And of course, it was not drawn by me. So I'm going to be running out of time shortly, but I might have time to have a look at one more story, and then I'll give you time for your questions. So I'm going to have a look at response in asthma, which is a field I worked in a lot. And the FDA uses 15% bronchodilation as a standard. That's really all you need to know from this particular slide. The FDA likes to classify you as a responder if you showed a 15% increase in FEV1, which is also known as VEMS, in French VEMS. And if your bronchodilation improved by 15%, then you're a calculated responder. This is an example of my own. So here is a trial I worked on, a crossover trial in asthma. There are 71 patients, and the forced expired volume in 1 second is being measured, and I'm going to use a percentage change in baseline. I don't like this measure, but I'm going to play the game the way the FDA plays it. So we have treatment A, and we have percentage change on treatment A, and we have percentage change on treatment B. And because this is a crossover trial, then actually I can measure each patient twice. And this is the response line of 15%. And I've classified patients as those who responded on both treatments, those who responded on neither, those who responded on B only, and those who responded on A only. And so it would seem here what we've got is we've got two particular treatments. The treatments have been tried once each on the patients. It's a crossover trial. So surely now we can say, well, there are some patients who responded genuinely on B and some who responded on A. However, I'm now going to reveal something else about these. These are two particular doses of the same treatment. One is 24 micrograms and one is 12 micrograms. And it is extremely, extremely implausible that a patient could respond under the lower treatment, under the weaker treatment, the one with a lower dose, and not on the higher dose. So why is it that we have in fact got these 8 patients down there? It's because we have overlooked the fact that patients vary greatly from occasion to occasion. And by only measuring them once, you don't really get a very good impression. And so my claim here is that the most likely explanation for these particular areas here is chance. And probably the situation is that 24 micrograms is rather more powerful, not very much more, but rather more powerful than 12 micrograms. But there is a huge amount of random scatter, in particular accentuated by 2 unforgivable sins, one using percentage change from baseline, which is a really bad measure, and the other one dichotomizing. So the real lessons, other things being equal, a high number needed to treat is indicative of a poorer treatment, but it is not a valid shortcut to studying variation. It's our duty as statisticians, and I speak from my particular profession, to understand and master the variation in the system. We should not naively overinterpret differentiation in observed response. Some of it may be genuine, but much of it may be simply random variation. In many, but not all cases, the task facing us will be to deliver better average medicine. The example of ulcer treatment is an example we should think about. In this connection, there's one big problem we continually overlook. The main source of variation in the system is not patients, it's doctors. And what we need to do is we need to have a look at how variation in the delivery of healthcare by doctors impacts on healthcare. And this is a quality matter. Finally, I'm going to leave you with a particular treatment that interests me, and that's a particular condition that interests me, and that's hay fever. I have hay fever, and so does my daughter. And my daughter, by the way, is now in her 30s, and I was interested when she told me what medicine she was taking to see, just out of interest, what the product information said about the medicine, and I noticed a personalised element. It says, giving this medicine to children. As a guide to a child of 9 years of age will weigh about 30 kilograms. So it wants you to think about this in terms of children's weight and their age. It says, do not give to children who weigh less than 30 kilograms. Do not give to children under 2 years. And if you're an adult and a child over 12 years, you can take 1 tablet once a day. But what about children who are aged 2 to 11 and who weigh more than 30 kilograms? The answer is 1 tablet once a day. And children of 2 to 11 years who weigh less than 30 kilograms, do not give this medicine. That, ladies and gentlemen, is the reality of personalised medicine. We have the opportunity to do it and we don't do it. Here, simply the same dose is given to adults and children. And I find it amazing that the pharmaceutical industry, which has been so slow to personalise medicine by sex or by body weight, should be so blinded by genetic and omics science that they think that this holds the key to the future of medicine. I'll leave you with this thought: the supply of truth always greatly exceeds the demand. Thank you very much.